Okay, good afternoon, everyone. Um, my name is Rob Duncan. Thank you very much for making the time for this exciting National Association of Plant Breeders webinar. Just so you know, this webinar will be recorded and available on the NAPB YouTube channel. I'm an associate professor and plant breeder at the University of Manitoba, and I'm the chair of the NAPB Education Committee, and I'm going to be your host today. You're going to have two moderators. Your moderators are going to be Dr. Bill Tracy. He is the Cliff Bar and Organic Valley Endowed Chair in Plant Breeding for Organic Ag at the University of Wisconsin-Madison. And he's also the interim chair for the NAPB Advocacy Committee. Your second moderator is Dr. Mikey Kantar, who is an assistant professor at the University of Hawaii, Manoa, and is also the chair of the Plant Breeding Coordinating Committee. So we have three excellent speakers today that we're very excited to have for your webinar entitled Regulating Breeding, Merging Technology and Technocrats. So those speakers are, in the order that they're going to speak, Dr. Jennifer Kuzma. She is the Goodnight NCGSK Foundation Distinguished Professor in the School of Public and International Affairs, and the co-founder and co-director of the Genetic Engineering and Society Center at NC State University. Dr. Kuzma will be speaking on the background of changes to the USDA oversight on gene editing and public governance. Our second speaker is Dr. David Bubeck, who is the research director for global breeding services for Corteva. David is also the vice president and incoming president, coming in August here, of NAPB. He will be speaking on examples and uses of gene editing and the regulatory implications towards commercialization of those products. And our final speaker is Dr. Heidi Kepler, and she is an associate professor in the Department of Agronomy at the University of Wisconsin-Madison, and is the faculty lead of Transformation and Gene Editing Technology Research and Development at the Wisconsin Crop Innovation Center at the University of Wisconsin. Dr. Kepler will be speaking on potential uses of gene editing and implications of revised regulations on genome editing and the potential effects on plant breeding application. Before we start, just a few details on the webinar. Each speaker will present for 10 minutes. After the speakers have completed their talks, we will open up the webinar for 30 minutes of questions and answers. Please keep your microphones muted at all times, and please type your questions throughout the talks and the question period in the Q&A box. We will make note of those questions and pass those on to our moderators. We will also be taking notes during the webinar and this recording, and use this information in the development of future information for the NAPB Advocacy Committee. So with that, thank you everyone again for attending, and I will turn it over to Dr. Kuzma.
Great. Can you all hear me and see my slides? Wonderful. So thank you for inviting me. It's really a pleasure to speak with you today, and I'm looking forward to seeing the NABP's efforts in this area in the future. So what I'm going to start with today, if I can advance my slides, let me see here. Aha, there we go. I'm going to start off with just a little history of the Coordinated Framework for the Regulation of Biotechnology. It was formed in 1986 as a policy document from the Office of Science and Technology Policy out of the White House. And some of the principles of the Coordinated Framework were that because biotechnology products don't present any new categories of risk, in other words, the risks are the same of kind, and that it's the product that should be the focus of regulation, not the process by which it's generated. That policy document set forth the principle that no new laws should be needed to regulate biotechnology. And this is in contrast to several other countries around the world, but the US approach is based on these existing laws that were on the book. So I'm not going to go over every box on this slide because it's complicated, and the Coordinated Framework is complicated. But I want to point in particularly to plants, so biotech products from plants or that are plants. The main federal agency that was assigned to regulate plants under the Coordinated Framework was the USDA, and they were to use this law that had been on the books since 1957, I believe, called the Federal Plant Pest Act. This was revised in the year 2000, bringing together the plant pest and the noxious weed authorities, and I'll talk a little bit about that later. But the regulatory hook for a long time was the fact that plants that were genetically engineered, were engineered with agrobacterium sequences to help move the DNA into the plant, and therefore it kind of provided a regulatory hook because agrobacterium is a plant pest. So that's what we'll be focusing on today is the USDA. I hope you can see my cursor there. But just to be aware, the EPA also plays a role in biotech plants under the Federal Insecticide, Rodenticide, and Fungicide Act, another old statute. And that's for plant incorporated protectants or pesticide-like proteins that you put into plants, like BT, for example. And the FDA also plays a role for food and feed. So that's just the overview. And again, it's because the next slide shows that the agrobacterium sequences are used to help move foreign DNA into plants. And I think most of you know that as very well as plant breeders, so I'm not going to belabor that point. However, now with gene editing on the next slide, we no longer have to use plant pest sequences necessarily, or if we do use them to get the gene editing machinery into the plant, they're often back-crossed out. So with the absence of plant pest sequences, a lot of the gene-edited plants initially were not coming under USDA's authority under the Plant Pest Act. So as you can see with gene editing, you have a nuclease that cuts the DNA, it makes a double-stranded break. If you don't have any template DNA, it'll repair itself, maybe causing a point mutation. If you do have a template DNA, you can target a point mutation in there, or you can even add completely new sequences, including foreign DNA, if you want to, by providing a longer template. So you can make deletions, small edits, large edits, or entirely new transgenes can be put in with gene editing. Generally, when people talk about gene editing, they're meaning SDN1 and 2, not necessarily putting in whole, entirely new genes from different species in, however. So this is the old USDA pathway for regulation. Is it a plant pest? Does it have plant pest DNA? And if the answer to that was yes, it would have to undergo either a permit or a notification field trial and then a petition for deregulation. If the answer to that was no, then what the USDA started doing in about the year 2010 is companies or public producers of biotech plants would put forth a letter to the USDA saying, hey, we didn't use any plant pest sequences, either with gene editing, they had been back-crossed out, or we were using vialistic and gene gun methods, and so am I regulated? And then the USDA would come back with a letter saying, generally, no, you do not fall under the Plant Pest Act. So that was the situation up until about 6 months ago. So again, since 2010, most of these products are going through the Am I Regulated process, a back and forth letter, all right? And that you can find these letters online, and here's an example of one for I think is a soybean resistant to cyst nematodes. You'll see sometimes they'll have a lot of confidential business information that has been deleted, like this example, and then the USDA will write back and say, based on the information you provided, you do not come under our authorities. Well, that has pretty much changed with the new secure rule. So I just want to, in historical context, the USDA has tried to update their regulations since 2008, because what happened was the Federal Plant Pest Act was merged with the Noxious Weed Act to form the Plant Pest or Protection Act, excuse me, that's a typo, I think, of 2000. So in 2008, they proposed a rule right at the end of the Bush II administration, and then Obama came in and they basically started over again. Again in 2017, Obama proposed a rule, and then Trump administration comes in. So there's a lot of political cycles to these regulatory changes. In both previous instances, though, USDA had tried to invoke their noxious weed authorities under the new Plant Protection Act, not just their plant pest authorities. However, the secure rule really focuses only on the plant pest authorities. And so I'm going to get back to this later. In my opinion, and this is my opinion, I think that's a weakness from a risk assessment standpoint. So here's some of the features of the new rule. Shift from the present of plant pest sequences to a plant pest risk. Now that's potentially a good thing. It makes it more risk focused and not so much of just the presence of plant pest DNA, which really doesn't mean that it's going to be a risk to the environment or human health. However, under this rule, there's a lot of categories of self or USDA confirmed exemptions. And I'll get to that in a second. And then those are things that have already been reviewed by USDA. So if it's gone through an MI regulated letter process or a previous permit or notification or deregulation, it's going to be grandfathered in. Or if it has the same plant host, the same trait, and the same mode of action, it will also be exempt. Gene editing will be exempt only though if it's a single base substitution, a host repair mutation, or if that change already exists in the gene pool of that plant species. So in other words, if it could be achieved by conventional breeding easily, it will be exempt. Multiple gene edits are not exempt, however. Now, an additional category that you can apply for exemption is if the trait could have otherwise been achieved for conventional breeding. So maybe it doesn't occur in the gene pool yet, but maybe you could have achieved it somehow through conventional breeding, like maybe a wide cross, a species-species cross, or a subspecies cross. And I think that's yet to be determined what kind of categories that's going to involve, but you have to apply for USDA for that kind of exemption. If it's not in the above exemptions, it's going to undergo something called a regulatory status review, and this will be a key point of decision making, and that'll be based on plant pest risk. Now, USDA expects 99% to stop at this regulatory status review. Weediness will be considered, but it's not a determinative risk for regulatory capture, which is a problem in some cases, as many, this is my opinion again, some of the risks that we're seeing with the first generation of genetically engineered crops are the overuse of herbicides leading to weed resistance or cross-pollination with wild relatives and weediness. But if there's no probable plant pest risk, it's going to be cleared and exempt from regulation. If a plant pest risk is suspected during the regulatory status review, USDA will conduct a plant pest risk assessment. And then if a risk is determined, it'll have to go through that whole permitting process. Now, all plants genetically or engineered or edited to produce a pharmaceutical or industrial substance must go straight to the permitting process according to this, the new rule. So here's a schematic of the new rule. And you see the exemptions up on top there, and I've already gone through those. And then you'll see, is it exempt? Yes. Well, you can request that USDA confirm that you're right that it's exempt, and then it'll go through. But there's also this self-exempt category. Now, USDA, I think, is going to publish a list if they confirm exemption, but there may be some gene-edited products or exempt products that go on the market that you could go through this self-determination pathway. I believe that's still the case. If it's not in those exempt categories, but otherwise achievable by conventional breeding, you can apply to the USDA for a new exempt category. Or if it doesn't fall into that, it's not achievable by conventional breeding, it'll go straight to this regulatory status review, or the question of whether it poses a plant pest risk will be asked. And then I already covered that. So some of the problems with transparency is that I have in red here, we think that there'll be a list for these ones that are USDA confirmed. We believe that there will be a list for the ones that undergo regulatory status review, but those are not going to go through Federal Register comment and rulemaking. The ones that will be if USDA makes a new exemption or if it requires a permit for a field trial, the draft finding of no significant impact and EA and that kind of thing. So there's some problems, I think, with public transparency with this approach. So I'm going to talk about the pros and cons, and this is one of my last slides, and I'm going to hurry it up a little because I'm at 10 minutes. So the pros and cons of the new rule is the pro, and these are my opinions now, not so much what's going on. The pros are that, and my timer is going off, so I'm going to dismiss that because I can hear it. It's bugging me. It may not be bugging you. So pros and cons of the Secure Act. The pros, shift to plant pest risk as a focus and away from just the presence of DNA, which doesn't necessarily indicate a risk per se. A tiered approach, I think it's good that we're exempting things with which we have had experience. And the provision of information for some of the categories and federal comment and rulemaking, public comment and rulemaking. In the Federal Register. The cons, I think a big con is failure to invoke the Noxious Weed Act, given the first generation of GM crops. And so there's a limited scope of risk issues considered, and that is whether the plant itself is a plant pest. Exemptions some of the exemptions have very little to do with product risk. As you all know, a single base pair change could really upregulate toxins in a plant or downregulate important things in a plant. Lack of public information for self-confirmed categories and perhaps some other categories, and lack of a formal assessment and comment period for the regulatory status review, which is going to be, I think, a big place where a lot of determinations are going to be made. So broader issues, it's not very inclusive. Key decisions will take place with no public stakeholder or external expert input. Problems with transparency. And now and involuntary consumer risk. So even if the risk of consuming genetically engineered or gene edited plants is infinitely small, when people are asked to take on an involuntary risk, something that they don't know about, they tend to view it as a higher risk. Voluntary risks like driving, flying, we take on and we know we're taking on that risk. It's going to be really difficult for people to find out what gene edited crops are on the market, where they are in their foods, and whether they're consuming them. And that is problematic from a consumer confidence standpoint. And then traceability is a huge issue, and I believe that the next speakers will talk more about that. But how will suppliers and exporters keep products segregated? Most gene edited foods are not going to have to be labeled unless they contain foreign DNA in the final food product. And so we're heading towards, I think, a lack of labeling, a lack of traceability, and probably more trade disputes that bring us all the way back to the 1990s and early 2000s, where we lost markets to the EU and other countries. And that's the end of my talk. I'm going to save the rest for everybody else. Those were just extra slides if I needed them. So thank you.
Thank you, Dr. Kuzma.
Sorry, I went a little bit over too.
Okay, thank you very much. Just a reminder to everyone, please start typing your questions in the Q&A box, and we can start organizing those and get those prepped and in queue for the Q&A session when all three speakers are done. And with that, I will turn it over to Dr. Bubeck.
Thank you, Rob. Everybody hear me okay? See my slides?
Yes.
All good? Okay. Very good. Thanks everybody for joining us today for this webinar. I think it's a timely and very pertinent one for today and the age we live in. I want to talk about implications in the private sector, and I'm gonna go through some specific examples of genome editing that we and/or others have chosen to pursue, and then talk about, from a multinational seed perspective of Corteva, some of the implications of the regulatory systems that we have in place. Dr. Guzma already talked quite a bit about Part 340 and the secure rule, so I'm going to touch on that a little bit and the implications, but I'll also talk a little bit about global trade implications to the private sector. So with that, you know, the whole you know, anytime we develop a product, there's a process that goes about, you know, from knowledge and technical knowledge of ideas of how to develop products for our customers. In the case of genome editing, you know, there are a lot of other implications on will the technology be accepted? What's the regulatory status? And then you go through kind of the normal routine of, you got to have a guide RNA design. You got to consider transformation and regeneration of the plants, of the species that you're talking about, some kind of molecular analysis that's imposed. And then, as always, in any kind of a breeding tool, no matter what it is, there are breeding implications to where do you put it in the process of your breeding effort. Do you do you do genome editing in every single thing that you're going to commercialize, or do you use back crossing or trait integration? And then how do you evaluate those potential products on that way up the pipeline to product commercialization? So a couple features here and considerations for the final product is that no DNA sequences from a different species is actually in the final product. So that is the way that we consider genome edited products, and I think most people are considering them. And then this implication that similar changes could have been obtained through traditional breeding, and that's already been mentioned. You know, I would have liked to see it go a little bit farther than this in description, is that, you know, eventually some generation of genome editing capability will go beyond what is normally thought of as a traditional breeding capacity. But right now, I think the way people are talking about it, it's going to be faster than what could have otherwise been achieved by traditional breeding. But is it a product that could have been similar and within the range of native variation that exists somewhere within that species or related subspecies? So I just want to talk about a couple specific products that what we've actually achieved. And one is around waxy corn. And the current waxy products that are in the marketplace have been there for you know, the product capacity capability has been there for many years. And that is, it's a partial, literally a native state, it's a partial deletion of the waxy gene, which enables us to get to nearly 100%, high 90-some% of amylopectin. And so the next generation waxy products that we've devised and actually have created in a very short period of the last few years is through CRISPR. And so it was a full deletion of the waxy of the gene that creates actually waxy. And a couple of intriguing things here from a breeding perspective is that many people observed that in the conventionally derived native waxy partial deletion, that sometimes, many times, there was actually a performance reduction from the native wild type, and that when you get a mix of amylopectin and amylose, that you actually have better yield. And from a classical breeding perspective, from the students that are on the line, you know, is it pleiotropy from being 100% amylopectin and a negative carryover to pleiotropy to the yield performance, or is it you know, is it literally physiological or pleiotropic, or is it actually a linkage drag based on where the gene is located in the genome? And so with CRISPR waxy, we were actually able to, you know, look at that in consideration. And the answer to the question is, I believe it's some of both. I think there's a little bit of a physiological effect and pleiotropic effect. And then there probably was actually a linkage drag that occurred in some of the waxys using back crossing and trait integration. So it's a little bit of both, I would say. Another thing that we are currently working on is northern leaf blight resistance. And this is more of an allele swap or an allele replacement, where we've identified a resistance gene to northern leaf blight. And we've used a genome editing approach to actually swap allele and have created resistant plants. This is in our product development pipeline, and we've talked about this trait. I think, again, here it's interesting to think about sources of disease resistance. And you can finally test using a CRISPR designed approach whether, you know, there are pleiotropic or linkage effects, because you are literally just dealing with the gene itself, and you're eliminating any potential chance for linkage drag to occur. So this is an interesting one that's in our pipeline. There are a number of others that we've talked about and talked about collaborating with others, but here's a number of other targeted mutation examples. Some of these, I think it's important to note that some of these are beneficial to the grower, to the farmer, and others of these are more beneficial to the consumer. And I think you can kind of sort through which of those would fit into each of those categories. And so I think the real benefits of genome editing, when they are realized by the consumers around the world, hopefully that will help us establish a bit more of the social license to practice the technology and the applications of genome editing as we go. Another thing that we're doing as Corteva is looking outside of the company and very open to collaborating as well as enabling. A big problem in Africa is maize lethal necrosis virus, and there aren't a plentiful supply of good genetic resistance sources for maize lethal necrosis, MLN, and so we've had a collaboration ongoing primarily with CIMMYT to identify the causal locus of MLN tolerance. So there is a pretty good source of resistance, as you can see in the picture there. And we are in the process of editing several CIMMYT inbreds to start a delivery of an MLN solution for African farmers. So another approach to, and a great example of what could be done through breeding, and could we do it faster with a much wider spread delivery to the right-based genetics at will, if you have a genome edited solution for MLN. So those are some of the examples, and I think in terms of the pathway towards a product, thinking about technology, those are kind of the easy ones for us as scientists. We have to have genes before we can edit them. We have to know something about those genes and the state by which will provide, of the state of the gene that'll provide a benefit to the performance of the plant. And then policy here. I've been involved in American Seed Trade Association working group that's around the Plant Breeding Innovation Working Group. And it's a team that's been assembled for more than 5 years that really is driving the question about how do we influence policy? How do we influence the changeover, the upgrade, the modernization of Part 340 that has already been described, to obtain the secure rule, which hopefully will provide a more clear path to the regulatory framework around genome editing. And then not the least of which is social license, and how do we work with end users, produce manufacturer association, food companies, and consumers as well, and consumer groups to establish that social license. So I'm going to skip over this one a little bit due to time, but it kind of shows in a little different way the process of where we were in the MI regulated process to the current Part 340 that's now been finalized, and I think she described it very well, so I'm not going to dwell on that one. I will comment a little bit more on the FDA and EPA. So just for everyone's information, there was a June 11th, 2019 executive order from the current administration that said the policy agencies in the US needed to upgrade, do regulatory streaming, and requested and actually had an executive order for them to update basically their rules around biotechnology, of which genome editing is considered part of what, primarily in the USDA camp, and that's the reason for the importance of Part 340, the secure rule. And then, but the FDA and EPA are not off the hook here either. So for example, what will the EPA do if they consider disease resistance mechanisms a part of a type of pest regulation? And if there's a gene product, or what if there's not even a gene product? What if it's a gene deletion that actually creates a resistance situation? So I think we're awaiting what the FDA and EPA regulations and rulemaking updates will look like. So I think that's, I wanted to point that out. Now, quickly, for us as a multinational organization, and we are largely involved in commodity crops that find their way around the world. So it's really important that we have an opportunity to influence not only domestic US and Canada regulatory framework, but in fact that we have some influence of the rest of the world framework as well. And until we have improved global alignment, products like waxy CRISPR corn are going to find some real limitations in how we can use them because of the global implications of trade. And so I think that's another real key point. So in the US, we have this coordinated framework between the 3 agencies. And I'm just going to list off a few of the other pertinent policy and regulatory triggers that are out there around the globe. Plants with novel traits, and the comments from the Canadian regulatory agencies about what, how traits are derived and are they actually novel and whether they should be regulated or not. And then the directive in the European community, and then the Cartagena Protocol on biosafety also makes a commentary on living modified organisms means any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. So how that affects genome editing specifically. So all these country regulations around the world are really important. And I think that the goal that we're all striving for, and this was a quote from the ISF, International Seed Federation, that plant varieties developed through the latest plant breeding methods should not be differentially regulated if they are similar or indistinguishable from varieties that could have been produced through through earlier plant breeding methods. And I think this is a tricky one for me because, I wish they would have gone a little farther in the statement because we always have improvements to breeding methods over the 30 years I've been involved in plant breeding. I think, there's a lot of tools, and to me, CRISPR and gene editing is just another tool, but how the world treats it today will be a really critical step as to whether we get to genome editing 10.0 or 20.0 if we never see our way out of 1.0 or 2.0 because there are such regulatory constraints. And so that's why I think it's really important for the NAPV to be involved in the policy direction, and for us to be able to have an influence in this space, I think is really important. So with that, I'm going to thank you, and I'll stop sharing so we can.
Thank you very much, David. I just want to remind everyone we're getting a few questions in the Q&A box. Please keep them coming because in approximately 10 minutes we'll start into that and with our moderator. So with that, Dr. Kepler, please.
Okay. Thank you. Well, I see we're running just a little bit behind, but that's okay because the speakers before me very nicely covered some things that I covered in my introduction part to my slides. So I think we'll be able to catch up and have covered the whole subject quite properly for a good discussion in just a little bit. So I'm going to talk a little bit about genome editing and the change in regulations and what that impact might mean for the public plant breeders. And oops. Okay. There we go. Sorry. So we already went over how CRISPR works and the different types of events that can come out of using editing. Insertions, deletions are the typical things, a few base pairs here and there, or large insertions or deletions, and so on. And then more interesting in terms of the current revised regulations and what may be coming in the future are, and as David just mentioned too, where you have cases where you want to do allele replacement or put in new gene sequences into an existing place of a removed gene, for example. And so when we think of kind of the different outcomes and what they might mean in terms of these new revised regulations, so we need to look at gene replacement or allele replacement types of outcomes, or promoter replacement, putting novel sequences in. So for example, in the first two categories, gene replacement and promoter replacement, this can become familiar territory with the whole area of cisgenics, where if you're replacing your gene or allele with sequences from crossable species, like in the cisgenic and intragenic cases, then you're more or less regulated under the cisgene type of system. And similarly for the promoter sequences too, where it gets interesting is in the novel sequences where you may be actually putting in a new function, then that tends to go toward, even though you did it with editing and you left no other parts behind other than your novel sequence, then that may go under a transgene type of regulatory route as outlined. And what about if you take sequences that have been deregulated, but maybe in another gene or another species, and insert those sequences? What is the impact with the current regulations? Then also, as we talked about the editing itself, the process still in many cases involves having integrating types of vectors with the CRISPR-Cas machinery, going in and programming the editing happening. And then if you have a grain crop, then you are able to segregate away the edit from the transgenics. And so that very much simplifies the regulatory process. But in clonal crops that aren't seed propagated, that becomes, even if you're able to edit, you've still got transgene sequences in there. And so it's going to go under a different stream of regulation. And that has pushed a lot of technology development of being able to have non-integrative editing where there are no vectors that are integrated. The molecular machinery is delivered, for example, as RNPs. You get the edit, and that's all that comes out through plant regeneration and into your next generation, your T1, T2 generations. So there are wide possibilities of the things that we can currently today do with editing and how they might be regulated under the new regulations. But even as David was saying, that the things that are in the future and that are coming, we don't know yet, and it'll be a continual landscape, I think, of modifying and improving the regulatory processes. So this was just an extra. If anybody had questions about cis and intragenics, I think we can jump that for now. And I wanted to just get to kind of the wrap-up slide so we can have enough time for a good discussion. And so from the plant breeding perspective, in the various applications, what might be some of the positive effects on these new regulatory changes? I think, potentially there'll be more options to use genome engineering or editing-based types of techniques for breeding research and crop improvement applications, because you'll have less limitations downstream in terms of regulations and costs and things, but also it allows the processes to become less expensive. That is, to actually make edited crops or make engineered crops as we lift some of the regulations. And then overall, the reduced regulatory constraints on a number of kind of simple edit types of crops that are coming out. The whole regulatory process for variety release commercialization becomes decreased in costs and impediments that were there, especially for kind of smaller, either public plant breeders or smaller private breeding companies. There's things underway that you'll have reduced IP related types of restrictions, which again, for public breeders and smaller private breeders is a benefit that's potentially going to happen. And improve public acceptance of the product if a number of the things that are edited are perceived as, we could have got this through different regular traditional breeding methods. We just had a quicker way to do it. Some of the negative effects, and this was already covered previously too, is that the things that are accepted here into the market without regulation might not be acceptable in foreign markets. And so where's your target for, if you're a plant breeder, where's your target for your crop to be grown? So that's a consideration. And for a while, there's still gray areas in the forms of editing that will or will not be regulated down the line. And, 1 or 2 base pairs is okay, 3 or 4 is questionable, new insertions and deletions. So I think, there's still stuff we're going to be hammering out in the years to come as the technology moves on. So with that, I will stop sharing so that we can have, and it looks like we have some time for discussion. Hopefully we've been able to catch up a little bit, and thank you all for attending.
Great. Thank you for all the speakers. I think really useful information. I want to thank Rob and Mikey as well for really pulling this all together. And Jody Scheffler, I should also say, who is our communications person at NAPB. So this all came together very quickly, and I'm really pleased also with the number of participants on the phone or online as well. So what we're going to do is I'm going to try to ask some questions and for the audience, and they are coming in. We have a few so far, and Mikey is assisting, keeping me aware of what questions are coming in. And so the first one that I have here is from Margaret Young, and this is to all the panelists. How deep/shallow is the sequencing to check for off-target deletions, etc.? Anybody want to try that one?
I think that one's for Dave, but I can comment that this was a problem with the hornless cattle, if you remember, that the ones who made the hornless cattle using a gene editing talons had initially checked using homology to the target site, and they had missed the insertion of one of their antibiotic resistance genes from the vector. So I think it's a really important question I don't know the answer to, but I think this one was directed at Dave. But just to remind people that this was an issue with the hornless cattle.
Yeah, we've had a lot of dialogue around this question, and I think there's a general attitude of we don't want to have to produce basically a full length DNA sequence for every single thing we commercialize. There have been review papers written on the likelihood of off-target effects. In fact, I was just a co-author on one that was published in Plant Phys not too long ago. And so the limited amount of off-target effects that occur is being noted, and also that those effects really don't have much of an impact on product in most cases. So yeah, we got to make sure that the non-species DNA is eliminated, but in terms of ensuring off-target effects, and we feel pretty good about effects that are going to be caught through any negative performance effect in our evaluations, because we're obviously going to evaluate potential products for years before we commercialize them. So any negative off-target effects, though they're low in their likelihood of occurrence, we feel like we're going to find. So I guess to answer the question, I don't think anybody wants to do a full sequence. Let's do an analysis of high density that catches, the obvious things, but I don't think we're overly concerned, and we don't want to do a full sequence just because of the expense.
Heidi, anything to add?
Yeah, it was the level of sequencing and finding off-target sequencing. There's good approaches for that for plants to really make sure that we're not concerned that there's excessive off-target hits. But sometimes I think that discussion gets more confused when we're talking about editing in the human side, where having off-target hits is a much more devastating potential type of a thing to occur than in plants if we get a ping here or there. So I think sometimes we focus maybe on trying to find total zero level of any off-target cutting happening in plants when it's maybe not really worth that same level of concern as you might have in a mammalian or human system.
Great, thanks. The first one I have is the next one I have is very simple. I don't have the asker, but can I get CRISPR waxy corn in the supermarket, Dave?
No, you can't. And
I thought it would be that simple.
Yeah, and the reason is because of the global trade issues that I mentioned. And it's a really interesting example because waxy is a very defined market. I think almost all waxy growers in the U.S. in Canada would grow under a contract. They wouldn't plant a waxy hybrid without knowing where the end use product is going to go. And yet, even with that, the byproducts that came out of the milling process go all over the world. And so we have, you know, we're a global, we respect global regulations. And as a result, we chose not to commercialize the product because we couldn't control where all the byproducts were going and the approval of those byproducts. So no, it is not on the market, but it could be, and it should be.
Thank you. Next question. Do you think that and again, directed, I guess, to the entire panel, we kind of heard about this in your talks already. Do you think that the rule will create, the new rule, the Part 340, will create similar public acceptance problems for genome editing as as we saw with the first generation of GM products?
I can take that one a little bit. So when you do, you know, large scale national surveys, social science, you know, using robust methodologies, you'll find maybe 20% that will reject GM foods. It's not, you know, the majority. About 40% will accept them depending on the benefit and the risks and how they're managed and that kind of thing. So it's not uniformly, the public's not uniformly against it. For those 20% and possibly even the 40% that are on the fence with regard to, you know, the benefits and the risks and how they're being managed, I don't think that the rule goes far enough to instill confidence, largely because of its transparency problems. There's a certain segment that you're never going to, you know, reach. They're always going to be against GM, whether it's cisgenic or transgenic. And in fact, there are some public perception studies that do show that on a whole, people are a little bit more accepting of cisgenic, but still not as accepting of cisgenic as naturally, conventionally bred, you know, like going to do a cross in the field or what have you. There seems to be a component of taking things into the laboratory for any step, whether it's gene editing or transgenesis or whatever, that makes people uncomfortable. So I don't think you're ever going to reach those people. However, I do think that there would be some things you could do to the rule and transparency and decision-making that could improve people's confidence that these risks are being properly managed. And one of the things to me is I think you all have to remember that the main risk that USDA is looking at is whether or not the plant itself is a plant pest. You know, they're not necessarily focusing the determination on risk of weediness or even on non-targets, really, unless it increases pests that are non-targets. So it's a pretty narrow set of risks that they're looking at. And I think they'd be better off doing a more comprehensive view of broad categories of ecological risks and benefits. And I do think that benefits should be in the equation in a lot of cases, rather than doing this in such a piecemeal approach, focusing on a narrow set of risks and having like multiple different pathways that are confusing to even experts like us that study the regulatory system. But that's my personal opinion. So I guess I think it remains to be seen, but a certain segment of the population is not going to distinguish between cis and transgenic. And it's going to become confusing to them, especially because gene editing itself can be used for putting in a novel sequence like Heidi mentioned, that doesn't occur necessarily in nature for synthetic biology or putting in transgenes. So I think it's all so confusing, and I'm not sure that people are going to distinguish. But not everybody's against, GM. So it's going to be a certain subset of the population.
Great. We're getting a lot of questions now.
A couple comments to add from me before you go on, Bill, is, I think the industry, the private sector is in a better place today to manage to work through the education. In the 90s, we didn't realize it was going to be a problem. And I think the industry, the private sector, I'll say at least, speak on their behalf, is we were pretty naive about whether the consumer would ultimately care or not. And really the primary benefit of most GMOs was for the farmer, for the producer, not particularly for the consumer necessarily. So there's the other thing. I think we need some examples of whether nutrition or flavor or stability of production. You know, do you want orange juice or not because of citrus greening? You know, I mean, there are some things that will have real consumer benefits. And I think we need to demonstrate the technologies through some of those to get social license too.
Okay. We're getting more questions than we can answer in the remaining time. Let me ask this one. Does pest and disease resistant traits via CRISPR have to go through the EPA or just USDA? For example, disease resistance that could have been introgressed from a wild species, will that now be totally deregulated?
So let me speak on a real specific one with the northern leaf blight. We believe that at the moment that would only have to go through the USDA based on the current secure rule and based on lack of additional guidance from the EPA and FDA. We think that the USDA has ultimate jurisdiction over that one. I commented a little bit on where is the EPA actually going to go on considering plant diseases in particular. Is it going to be considered a pest regulation aspect? You know, what actually is the active, is there an active ingredient that controls the disease pathogen? And so I'm waiting to see what the EPA says on their, you know, new rules and new regulations revised. But I'm hopeful, and I believe it should be purely the USDA space, in my opinion.
Yeah, agree.
Let's see. For all speakers, is the controversy about the intellectual property of CRISPR-Cas technology something that will be considered in the registration process? Not sure about that one.
What's the question?
For all speakers, is the controversy about the intellectual property aspects of CRISPR, meaning what, I guess, who owns CRISPR-Cas technology, something that will affect the registration process? That's what the question is. Maybe,
Well,
go ahead, Heidi.
Yeah, I don't think it'll affect the just like any kind of intellectual property. I know they're fighting over who owns the licensing ability to that, but it would just, you know, as long as you've covered your bases in terms of licensing to be using that technology, I don't think that should affect the process of commercialization.
Okay.
I think one way that it does affect it is the amount of information that is claimed as confidential business information when you submit a regulatory package. So if you submit a package, you can claim a certain amount, and I'm sure David knows way more about this than I do. You can claim something as confidential business information in order to protect that intellectual property. It's a problem with public transparency because if you look at some of these "Am I regulated?" letters, you can't even tell what they're trying to do in some cases. In other cases, they're very transparent. So I would just say, you know, if you do, it's a balance between protecting intellectual property and being transparent with the public. I know that might not have been where your question was going, but I just thought I'd throw that in there.
Why is mutagenesis considered generally recognized as safe, but not editing in food species?
Yeah, why? Just people are just more comfortable with what was done in the past, and you know, on a scientific basis, it's not safer, or you know, they're equivalent, at least editing or directing it. So it seems to me that it's, people just feel like mutagenesis has been used for a long time and nobody's that they know of died from it.
And I do think the USDA rule was in part to address that by the single. I know that with mutagenesis you get multiple mutations, but I think the USDA was trying to address that in 340 by exempting out some of these categories of single base pair changes. Mm-hmm. However, they only left it to one change. So if you have multiple changes, I think you have to go through that other category where you have to ask for an exemption. But if it's otherwise achievable by conventional breeding, and you might be able to be pretty quickly exempt. So I think they're trying to address that. I guess it'll remain to be seen how exactly they make those determinations for other exemptions, is sort of where I think it's going. So they're trying to address that risk, like that the risk is the same in kind of thing. I'm not sure they've gotten all the way there yet, but they're trying to address it.
I will say that just recently, somewhere in Europe, it might have been France, was talking about actually, at least from organic agriculture, banning mutagenesis products, which would then create trade problems between the US and France in terms of organic.
Well, the Organic Standards Board has banned gene editing, correct? For those of you who are organic breeders, they've made a couple determinations about gene editing, that any kind of gene editing is not allowed. And so that's another problem in trying to segregate gene edited products from organic products as well.
I'm going to turn this over to Mikey because I actually have a 1 o'clock call. My entire day is back-to-back calls. I would like to thank everyone again. And I would like to also say to our attendees that the process that we're going to go about here is we're going to gather this information and other information, and that's my point, is you can send me comments at my wisconsin.edu address. You can look me up at University of Wisconsin-Madison and send me comments. That material is going to be brought together, hopefully to create a white paper on this issue to be discussed at the advocacy committee meeting of the NAPB in, you remember the dates for that, Dave?
Mid-August.
I remember those dates. Well, I'm going to sign off, and Mikey will ask the last question, and I want to thank everybody again.
Thank you, Bill. Yeah, so I guess for our last question, I apologize that we weren't able to get to everyone's questions. We really appreciate both the attendance and the participation. But I guess if we could just go around, what do you think gene editing will look like in the next in the next 5 to 10 years, in terms of regulation and the products that we're going to see, and who will be producing these products in the market? And I guess if we could go in the maybe in the same order that the speakers spoke. So first Jennifer, then David, and then Heidi. That would be great. And thank you again so much.
So where do I think the market's going to go? I do think that gene edited products are going to be out there and everywhere. And I think that despite some companies are going to do things, and I do think there's been a movement in a lot of biotech companies and biotech seed companies to be more responsible and responsive to the public. And so, I appreciate that some companies are not going to want to do this unless it's okay all over the world. But I do think there are other companies that are going to put these things out there, possibly. And I do think that in 10 years, they're going to be everywhere. And I think it's just going to be a matter if we repeat the history of the first generation from a public confidence standpoint, and perhaps a risk standpoint when it comes to some risks like herbicide tolerance and such. I think that it remains to be seen.
Yeah, I would add that I commented that you have to have a gene to edit a gene. You have to know something about the gene to edit it. So there are a limited number, I think, of genes and product opportunities in the near future. I think because of the global trade situation, you're going to have a very defined and understood market of where the product is going to go. I think the multinational commodity organizations are going to have to sort through with the grain traders. In the case of commodity grains, for example, how are we going to deal with this? And so we're talking to grain traders right along. I do think the U.S. in general, I'm going to make a general comment about the U.S., is going to be a respecter of everybody's regulatory climates. Whereas other certain countries, I won't name, but might not be as respectful of the rest of the regulatory climate around the world, and or the crops and products that they raise stay within country. So they may establish their own rules and not feel a compelling need to have rules that are, homologous and simultaneous approval with other countries, because it becomes a trade opportunity. So anyway, that's just, I think I'm concerned about the limited growth of the technology because of some of the regulations that will prevent us from really driving it to the next generations of technology use.
Yeah, and I guess I'll chime in then on the kind of second part of the question was about, what's the gene editing technologies of the future? And it's hard to predict right now, because it's amazing how much advancement there has been with this technology. You read new papers every day of new and improved or new things that we can do with gene editing, and it's so hard to even keep up now. So to imagine where we'll be with editing technologies even in 5 years, it is hard to think about, because if you look at what's happened since really the gene editing with the CRISPR-Cas system anyway exploded around 2012 or 13, and all of the advancements in a relatively short amount of time in the technology itself.
Well, thank you guys so much. I know we've reached the end of the hour. Thank you so much to the panelists. Thank you so much to Rob for organizing everything. Wonderful talks, and thank you for all the participation. And we look forward to hearing more.
Thank you.
Mikey.
Bye.
Bye.
Thank you, everyone.